The second half of the process of protein synthesis is called translation. Translation takes place within the ribosomes that are within the cell. You can see written down here in blue is the same chain of letters that existed when we were talking about transcription. This is your functional messenger RNA. The introns have been cut out and the exons have been spliced together. That mRNA left the nucleus and has now shown up at one of the ribosomes. It is going to occupy what is called the small subunit of the ribosome. The ribosome is made up of rRNA, or ribosomal RNA. So we've had mRNA. We've now established that the ribosome is made of rRNA. The only other RNA that we have left is called tRNA, and the T is what is pictured here. The key to the tRNA, or transfer RNA, is this little group of letters down here in the bottom. If we look at the next screen, you'll focus in specifically on the tRNA. You can see that at the bottom there are those three letters called the anticodon. So the three bases at the tip of the tRNA, the anticodon, must match up with the letters of the mRNA. In other words, here is a group of three letters of mRNA, and that group of three letters must match up with this group of three letters. This group of three letters within the mRNA is referred to as the codon, and this group of three letters within the tRNA is the anticodon. Codon must match up with anticodon. At the other end of the tRNA is an amino acid. Now this is significant. I said at the very beginning of this entire process that the whole point of protein synthesis is to make a protein. Well, what are proteins made of? Their monomers are amino acids. So if we're going to make a protein, we need the small sections of amino acids to get linked together in order to make that protein. Translation is when all of that is going to occur. So if we look at this particular picture right here, here is supposed to show you in the background here in gray, that's the ribosome. And on top, this funny shape up here is called the large subunit. And at the very bottom, the smaller portion is called the small subunit. And you can see that the mRNA lined up along the bottom is occupying that small subunit, and then the large subunit is being filled with tRNA molecules. And if we look at this one right here, this tRNA has an anticodon of GAC. So it is the only tRNA that can match up with this codon of CUG. The other significant thing here is that this tRNA is carrying an amino acid, and not just any amino acid, a specific one. You can see the LEU up here just simply stands for leucine, which is the name of an amino acid. Here this tRNA, with its AGG anticodon, is the only one that can match up with this UCC codon. It too is carrying an amino acid, serine. And this one's carrying an amino acid, tyrosine. And each time these tRNAs line up next to each other, a chemical reaction occurs that links together the amino acids. And so what you're seeing here is a growing chain of amino acids. And this is just an assembly line type process. When this tRNA has given its amino acid, it leaves, and then there's room for another tRNA to come in. And then the next one will leave, and there's room for another one to come in. So you will work your way down this whole chain of mRNA until the last letters are used up, each with a tRNA anticodon coming in to match up with the mRNA's codon. And so in the end, you will create this growing chain of amino acids, or polypeptide, because the bond that forms between them is called a peptide bond. And ultimately, once that polypeptide chain of amino acids ends up twisting and folding into the proper shape, your body will have made the correct protein. As I mentioned, the amino acids that are being carried by tRNA are very specific. Each tRNA only carries the correct amino acid. This chart is a listing of all the different amino acids. You can see phenylalanine and leucine and isoleucine and valine and so on. And so if we look at an example here, here is a strand of DNA. Now I've just typed out the antisense portion of it. So TAC, CTT, GGA, ACT. During transcription, what we talked about earlier is that the RNA polymerase enzyme converts these letters into mRNA codons. And so that's what you're seeing here in this column. AUG, there's one codon. GAA, there's another codon. You can see they match up. T with A with U, C with G. And so eventually you form that mRNA strand that leaves the nucleus and goes to the small subunit of the ribosome. While there, a tRNA with a specific anticodon matches up with it. UAC is the only thing that can match up with AUG. And CUU is the only thing that can match up with GAA, and so on down the list. This tRNA then is carrying an amino acid, a specific one. It carries the same amino acid every time. UAC always carries the same thing. CUU always carries the same amino acid. And so over here, you can figure out which amino acid by using the chart. Now this gets screwed up a lot, and I'll show you why. You would think that that chart would be based on these letters right here. This is the item that is actually carrying the amino acid, so you would think, okay, the chart is going to be based on these letters, and you would look up UAC to figure out that the amino acid would be methionine. But that's actually wrong. For whatever reason, and I can't tell you why, it is the mRNA letters, the codons, that the chart is based on. So if we look up AUG on the chart, notice it says first base, second base, third base. If I look up AUG, the first base is A. Here it is. The second base is U, so bring together the A and the U. And then the third base is G. Bring it all the way over here. You can see AUG. We've got methionine. If we look up the second one, again, we use this column. GAA, GAA, we've got glutamic acid. Glutamic acid. And then CCU would give us proline, and UGA gives us stop. In other words, the ribosome has to know when its protein is done. How does it know when it's done? Well, every protein that your body makes is going to end with what's called a stop codon. They all start with a methionine, which is known as the start, and they all end with a stop codon. Now, we've got 1, 2, 3 amino acids in our chain here, and then I have a stop. That's not realistic. You would have hundreds, if not thousands, of amino acids, and then eventually there would be a stop codon so that the ribosome would know, okay, my job is done, it's time to end. And if you look at the chart here, there is more than one way to stop. UAA gives you a stop. UAG gives you a stop. UGA also gives you a stop. Those are the three stop codons. There is only one start codon, AUG. So everything starts with AUG. Everything ends with a stop. And now it's time to stop.